Hey Detlev, really
Hey,
good to have you on here. Thank you so much for jumping on with me. I was having a conversation with a mutual friend between us, Tracy, a couple of weeks ago.
yes.
And Tracy was amazing to talk to. And she was giving me a lot of detail on her background and her history, her autoimmune disease, and just the difficulty that she's had during her lifetime with autoimmune diseases. And then finally coming across TSO. And we were having this great discussion. It was fantastic. And then at the end of that, we were chatting after the video was done. And she mentioned your name and she said that I should reach out and get in touch. And me and you should have a conversation, have a chat about it. So thank you so much. It's awesome to be able to have a conversation with you. And I'm hoping that whoever might end up watching this, they get something positive out of it as well. But if you're happy, I'd be really intrigued to know a little bit about your background and your history and just like the years ago, how you got into the space and when then we can go into a bit more detail after that.
Yes. Well, this is a pretty strange story because I'm not a medical researcher by training. Actually, I'm a tooling engineer with an additional background in economy. And as such, I was working on in the laboratory with steel particles. And I always was very much bothered about microscope slides.
Yeah.
And microscope slides tend to stick in an humid environment.
Yep.
And I developed a small little thing to produce a microscope slide that causes that they don't stick together anymore. And I proposed this to the manufacturer, which was a company that was working in the laboratory field, in the glass world,
Mm-hmm.
and in the pressure relief systems.
Mm-hmm.
And they hired me right away from the university.
So this
And
you were
that's
doing
how
this
I
all at university, were you?
oh yes,
Yeah.
yes, and that's how I got into the medical business. And later on, I went more deep into the pressure relief systems, which is applied in wound care.
Yep.
And in wound care, I became the marketing manager of another company in wound care. And as such, I used to invite key persons, key leaders,
Yep.
doctors, about once a year to so-called brainstorming weekends. And I asked them the question, what is your biggest problem at the moment?
Mm-hmm.
And does anyone have an idea how we can solve this?
Mm-hmm.
And the biggest problem in wound care at that time was the the increased resistance against antibiotics for the treatment of infected wounds
Mm-hmm.
and the removal of dead tissue out of a chronic wounds so it can heal again. So that's
Mm-hmm.
called the debridement. And then we just thought, where can I find the most hazardous environment because of the infections?
Mm-hmm.
And that was in dead tissue
Yep.
in nature, dead animals.
Yep.
And there you can find maggots. And maggots of the blowfly have developed over thousands of years a system that protects them
Mm-hmm.
against the strange bacteria.
Mm-hmm.
And they produce kind of secretion in their mouth that they spit into the wound. And this dissolves the necrotic tissue and attacks the bacteria, the bad bacteria.
So this is the maggots.
That's the
The
maggots of the
maggots have.
blowfly.
Yep.
Yes.
Yep.
And that has been reported to be used in wound care thousands of years ago
Mm-hmm.
already, but it got kind of forgotten in medicine.
So is this
So
the
I,
idea? Is this the idea where people would be using maggots on an open wound where there's sort of
yes,
dead tissue around that wound? The maggots come
yes,
in, they clear up the dead tissue,
yes,
and they leave the good
yes,
stuff, right? Yep.
yes.
Yep.
So I proposed to my employer, why not going for maggots? Develop a process that can be applied intentionally
Yep.
in wound care and make it a product. And they thought this guy is nuts, and that nobody would ever do this. And that's the time where I decided to quit my job and set up my first own company.
Wow,
That was
cool.
called Biomonde Company, and I found some biologists. I went to the
Hmm.
local university here in Hamburg and found a student who was uh, writing his master on the fly,
Yep.
and I hired him right away, and then I found some doctors to join the team, and we
Wow.
developed a process to produce maggots of a certain blowfly in a sterile environment
Hmm.
that it can be approved by medical regulators.
So are we talking
And
like the FDA type thing?
yeah, well, that's FDA approval. Here
Yep.
in Europe, it is EMA approval.
EMA.
And
Yep.
it also requests certain local approvals by country
Hmm.
agencies.
Hmm.
And that was when we were the very first team worldwide that was able to get a full approval on a live animal for treatment of human wounds.
Wow. Wow. That's
And
and that was purely around the use of maggots from
yes,
this
yes,
particular blowfly. Yep.
yes.
Wow.
It
So
was
what did it what did a general treatment look like when you got this approved? What was this like? What? Give me an example of what someone would
how
be
it
come
looks
in the
like.
hospital, whatever. Yeah. Yeah.
Oh,
What
well,
is it?
yeah. Well, in order to make the application easy, we took some kind of a gauze,
Yep.
and we put the maggots into the gauze, and
Yep.
then clap it and seal the gauze.
Yep.
And that product we called a biobag.
Right.
And this, you didn't see the maggots anymore in this. The gauze was covering the maggots
Yep.
visually. And
Yep.
then you just take the kind of a cushion and put it into the wound and put a bandage around the wound.
Wow.
And inside this cushion, the maggot produces a secretion that flows through the gauze into the wound, dissolves the necrotic tissue, and the maggots inside the bag, they can suck it up. And that way they clean the wound precisely. They just remove the dead tissue and leave the healthy tissue alone, which the surgeon can never do. Surgeons doing a debridement always need to make a deep cut into the wound to get the necrotic tissue off.
Wow.
And the maggots can do this precisely better.
Wow.
And after 2 or 3 days, maggots inside this cushion grow, and then you remove the cushion and put it in the garbage outside in nature. They turn into flies and fly away. And the
Wow.
wound is
Wow.
clean of debridement. The clean had a clean debridement and is usually free of further infections and can heal well.
So
And
the secretion from the maggots
yeah,
on like day one, that has no detrimental effect on the healthy skin or the
yeah,
healthy wound per
yes,
se.
yes,
It literally
yes,
will just go in and attack the
yes,
bad. Wow. And then the maggots will actually and it will break it down, and the maggots basically just suck it up.
yes.
Wow, that's really cool.
That's a perfect solution made by
Yeah,
nature and developed over thousands of years.
it's amazing.
And people certainly try to copy it by synthetic molecules.
Yep.
People try to identify which molecule would be responsible for doing this phenomenal approach, but it is absolutely impossible to say it's just one molecule. Nature is too complex for that.
Yeah.
We identified up to 500 different molecules in the secretion of the maggot
Wow.
that is changing all the time depending on the environment they are placed in. So this is something you definitely can never copy synthetically.
Wow.
So still until today, people are using maggots in special wound care centers all over the world now.
Yeah.
And it was certainly an exotic approach that was published in many magazines and TV shows and so on, and got well known. And when I saw that nature offers such a perfect solution for a problem we cannot solve on our own, I thought there must be more out there in nature that you can convert into a therapy. And since I'm not a doctor, my approach was more a logical approach. I just went into the international database, medical database like PubMed,
Yep.
and I searched indications of diseases that are still known to be uncurable until today. And what you first look at, if you're not a doctor, is where can you find these diseases? Epidemiological data. And
Yep.
when I came to Crohn's disease, the inflammatory bowel diseases like Crohn's disease and colitis, ulcerative
Yep.
colitis,
Yep.
I found that you can only see them or find them in so-called developed countries
Yep.
and not in so-called underdeveloped countries.
Yep.
And that was the first hint. And then I searched more with the keywords of Crohn's disease and developed countries and undeveloped countries. And certainly with another keyword, hygiene. Because the main difference between developed countries and undeveloped countries is a very high standard of hygiene in our developed countries.
Yep.
And then I found a tiny little article written by Dr. Joel Weinstock. He, at that time, was the doctor-in-chief at Iowa University Center of Digestive Diseases.
Yep.
And he had a hypothesis that the high standards of hygiene might be a reason why we can see so many Crohn's disease, inflammatory bowel diseases in general. And I contacted Joel Weinstock and I told him about what I did with the maggots.
With Biomonde. Yep.
Yeah. And he said, well, I had a similar strange idea like you with your maggots. I was thinking about using a parasite to bring intentionally into the patient and see how this can do immunologically to the patient and the disease. And this indeed was a similar strange idea that like I had with the maggot therapy. And then I told him, well, why don't you come over here to my laboratory, to my company, and see what I did with the maggots? And he and two of his colleagues, Bob Summers, unfortunately deceased a few years ago, and David Elliott, came over to Germany, and they looked at what I did with the maggots, and I convinced them that we could build up a team together and see how we can do this with the parasite.
Now this is all happening around 1995. Is that right? This is
yes, around that time.
Around this time. Yep.
And then we had to find a suitable parasite. And again, as a tooling engineer, that's not my area of expertise. So we contacted a lot of parasitologists all over the world. And one parasitologist and microbiologist, he at the time was a senior biologist at the United States Department of Agriculture in the United States. And he had some research done with Trichuris Suis, the pig whipworm.
Or TSO, right? TSO,
yes,
for sure.
or TSO.
Yep.
And he was kind of a very good specialist of Trichuris Suis, more from the other perspective, because Trichuris Suis is a parasite that farmers don't like to have on their pig because it slows down the growth of the pigs. And that's something farmers don't like to have. So United States.
He was looking at it from the perspective of how do I get rid of them?
Yes, exactly. But he proposed to use Trichuris Suis because it filled some certain very important safety features. Because
Mm-hmm.
the parasite we want to use should not multiply in humans. It should
Mm-hmm.
not walk around other organs. It should stay in the gut,
Mm-hmm.
and it should be easy or manageable to produce commercially.
Mm-hmm.
And that was Trichuris Suis.
Mm-hmm.
And then,
So while you were going through this process and you were looking at different parasites and that sort of thing, were there many other ones that you came across that you thought could be a really good option as well,
oh
or
yes,
did you?
absolutely,
So
absolutely.
what was this process like when you around 1995 you were having this conversation with Professor John? How many other options were you looking at, and how long did it take you to actually come across TSO?
Well, actually, it was no other option. No, we had no other option than TSO because all other parasites had disadvantages that we don't want to have at that time. Because when you do such an approach, you've got to be really, really careful that the
Mm-hmm.
patients don't get hurt.
Mm-hmm.
And all our other parasites that we looked at had some kind of disadvantages. Either they are not staying in the area we want them to stay, or
Yep.
they have side effects that if you overdose them, people can really get serious diseases. All this kind of stuff we needed to eliminate. And TSO, Trichuris Suis, was really the best choice.
Yep. Yep. And just to double back on that, then the three key areas that when you were looking at from a safety perspective for something like this to go ahead was that you didn't want the parasite to move; it had to come in, had to stay in the same place. You didn't want it to multiply and grow and whatever else, and you didn't want the side effects for the host to be so significant that they would get sick in other means. They're
Yes,
basically the
yes.
key criteria, right? Okay, cool.
Yes,
Yep,
because
perfect.
Trichuris Suis in humans is kind of self-limiting.
Yep.
Trichuris Suis, even in the natural host, they settle down at the mucosa
Mm-hmm.
in the gut around the cecum area, and only in
Yep.
this area.
Yep.
And if you take more eggs, ingest more eggs, then they can only attach in a number that they have space in this area. If there are
Yep.
more that don't have the space to attach, they go through the gut and get out of the body immediately again.
Yep.
So that's another very fabulous safety feature for us.
Yep.
And safety was the highest priority.
Yep.
Efficacy was the second highest.
Yep.
So if we would have chosen another parasite that can do harm, but is more efficient, then we would have to accept side effects, and that would make the drug approval or the approval process very, very difficult. Like
Yep.
we can see now with other parasites. In the meantime, some people are also using other parasites, like the red tapeworm or the hookworm, or even the human Trichuris, Trichuris trichura, and that is very, very difficult to get approved by regulators because of the other features they have. They
So
work.
yeah, that's a really interesting point because for myself, I have recently started trying to use Necator americanus, so
Hmm.
NA. Um, but my understanding is that they basically tick all the three key boxes as well, especially being a human worm as well.
Yes.
So I am intrigued to obviously go through this conversation with you around TSO because it is um, quite a different worm in that sense.
Yes, well, it works. No question
Yep.
about this. But
Yep.
if when you want to get something approved, regulatory approved, you have
Mm-hmm.
to stay on the regulations of manufacturing too, like the
Yep.
GMP, like good
Yep.
manufacturing process. And for this type of product, it requires that you have a controlled environment from start to finish
Mm-hmm.
until the patient drinks it, and until from the starting point where you get it from the animal. And
Yep.
the animals in our case, pigs, need to be GMP pigs. So they need to be born in a sterile environment,
Yeah.
mainly by a Caesar's cuts, for example. That would be even better then. They need to be kept in a sterile environment, and they need to be inspected by veterinaries and microbiologists all the time. And even if one pig catches a cold, we have to discard this pig. You can't do this with humans because
No.
Nicata americanus is only growing in humans.
That's right.
So you have to get your product from another human. And we won't be allowed to run the farm with humans.
Yeah. Yeah, which I guess is probably one of the problems.
That's one
so
example.
to take it, yeah. So
That's one.
to step back then, so you and Professor John, you've come across TSO. He's checked out your lab. What sort of happened from there?
Then when we get it from sterile grown pigs under GMP environment, then we have eggs, and these eggs have a very, very strong shell made of chitin, and this very strong shell is resistant against a lot of acids. So what we are basically doing is we are treating these eggs when we get the worms out of the pig or the eggs out of the pig. Then we are treating them with different kind of chemicals to make it sterile. Then they are washed several times. They are brought into a very acid environment, and they survive all this, and they come out almost sterile. And then we can transfer them into a suspension, which is a phosphate buffered saline solution that is very similar to the solution that are used in hospitals for infusions, for example. So it's a very, very neutral fluid and has no harm in it at all. And then it becomes a drink. And the patients we're treating, they just drink such a small bottle that is halfway filled up with 15 milliliter of this phosphate buffered saline solution and contains a certain number of eggs. And when they drink these eggs are passing the stomach acid area, which is very acid, and even survive this. And compared to probiotics, for example, most probiotics you drink are completely destroyed in this very acid area. And when only very little quantities are reaching the gut, while TSO with this strong chitin shell survive it completely and safely arrive at the cecum area in the gut attached to the mucosa. And then the tiny little worms of larvae stage 1 are hatching from the eggs, and they dig into the crypt of the guts.
So as they're doing that journey down, and they go through the stomach, and they come onto the mucosal layer, how do they know? Like, what? I mean, maybe it's a stupid question, but how do they know to stop there and attach, and then what? And then what's actually triggering them to turn to larvae?
And that's how the way they trigger this is by a very, very large jump in pH values.
So the pH level in the saline solution that you're producing when you produce the drink for people to take,
yes,
that's around 2.5. Is that right? pH 2.5.
Yes, that's right.
Yep.
Yes.
Yep.
2.4 is the pH value Coca-Cola has, and we are around the same value, 2.4, 2.5.
Right.
And that's easily drinkable and has no problems at all.
Yep.
And
so that's what's actually happening then.
Yes.
So you drink it, and then it's that pH difference that's actually triggering them to hatch, per se.
Yes. The pig's gut, where they developed, where the worms are coming from, has a pH value of approximately 8. And when the eggs are shed by the pig into the soil has an average pH by 5, approximately, depending on the area where the pigs are.
Yeah.
And the eggs can only develop outside the pig. So they have to stay in the soil for a certain time to
Mm-hmm.
embryonate.
Mm-hmm.
And when we pick them up, we clean them, and then we kind of sterilize them and bring them into the pH 2.5 slowly.
Yeah.
The process when we pick up the eggs and adjust the pH value is in very slow and in small steps. So there is no sudden pH jump.
Yeah,
Okay?
because if there's a sudden pH jump, presumably that would trigger them to.
Yes, yes. And
So how long would that process take from when you pick them up from the ground and you get them to the point where they're in a solution ready to drink by a patient? What's that time? What's
they
that
need
time frame?
approximately 100 days to develop
Wow.
outside the pig's body to develop
Wow.
into a viable egg that contains a small piece of larvae.
So it's a really long process. It's not a
Yes,
simple.
it is
It's
absolutely.
it's a
When we start the process with the pigs, when the pigs get infected with trichuris, intentionally infected,
yeah.
um, then it takes approximately 60 days until the given eggs into the pig develop into worms, and the worms start laying eggs.
Mm-hmm.
And then after that happened, it takes about 100 days outside the pig's body to develop into a larvae inside the egg. And then
Yep.
we start the process of sterilization and produce the fluid and make a drinkable product out of it. And that
Wow.
takes another 3-4 months, for example, because
Wow.
it requires a lot of testing in between to make sure we have no certain viruses on there and there are no bacteria on there and all this kind of stuff. This is a permanent testing and looking if it's okay. And if there is only one kind of a bacteria in there or any other thing that we don't want to have, we have to discard the product and run a new
Wow.
batch.
So how many eggs are you able to harvest on average from one pig once they're infected?
This
What
differs. This
it
differs.
differs, yeah.
It is not a fixed number. Sometimes when we are using a batch of 10 pigs, for example, it can
Mm-hmm.
easily happen that sometimes 8 pigs are shedding the eggs and we'll
Mm-hmm.
get the Trichuris worm, but it can also happen that sometimes we have none of them. So we still, until today, we don't know why some animals have a resistancy that they don't shed eggs, or some have. So this is
Yeah.
something we need to work on to find out why this happens and when it happens.
So what would a good haul of eggs be? When
Well,
like what's a good amount look like?
um,
Obviously, it varies a lot, but if, yeah, you get it. You pick some worms up or some eggs up, and you're like, oh, that was a great. We got a good stash today. What does that look like?
uh, we have well the eggs are in a size of 30 to 50 microns, so they are
Yep.
not visible by normal eyesight.
Yep.
Even if you
Yep.
look into the vial where the fluid is in and hold it against the light, you can't see anything. You just
Yep.
need a minimum of 100 times magnification to see what's in there.
To actually say, yeah. So it's almost impossible to tell exactly in one big batch how many you're actually getting. Yeah.
In one big batch of 10 pigs, we might get something, an average from number of between 200 million to 500 million eggs.
Wow. Yep. So, that's a lot.
Yes, it
It's
is a
almost
lot.
unfathomable how many it
It
is.
is a lot.
Yeah.
And it
Yeah.
is a lot of material to check on and to analyze and to examine. So with a long process, because we have to do
Yep.
it manually at the moment. Of course, it is possible to design machines that are doing all this stuff much more fast and more accurate, like the manual examination under the microscope and all the kind of stuff. But that requires
Oh,
a hell lot of money.
yeah.
That was something like I guess something like $500 million US dollars
Wow.
to set up some kind of machinery for this.
Wow.
And it is also a very, very cost-intensive process to get it approved by regulators. For example, if there is a new development of a molecule, a new medicine for any kind of disease, a novelty, for example, then you have to run, you have to make the development at first, then you have to make the toxicity studies and clinical phase 1, 2, 3, and you have to do the clinical studies twice, that they can be comparable if you have a novelty that has never been there before. And the cost for such a regulatory approval of a new treatment can easily reach between 600 million to 1 billion US dollars.
Yes. And quite a long time frame too, right?
Yes,
well, this is over a number of years. This is,
and actually, this is the reason why so many novelties in drug development are so expensive. It's not because the process to produce it is so expensive if you have everything in place. It is you have only, when you have a patent on it, you only have like 15 years, 20 years maximum time to get a return of investment of $1 billion. That is what makes new drugs or new treatments so expensive.
Yeah, but that's the only way to get mass.
And
which is
here we have another big problem because the big pharma companies, they have the financial ability to spend this money. But this kind of a very exotic approach, like using maggots or a parasite, nobody dares to touch it from the big pharmaceutical industry.
Too much hassle.
Too different to what they are used to.
And that is a great lead in, actually, because one of the things I wanted to talk to you about on the back of this was Overmed, and the process that you went through with that, because from what I understand, you originally did partner to try and go through the FDA type approval to get something out there. Are you happy to have a wee rundown on that? Because I think this is a really interesting part of the history of what you've been doing.
Yes, I have. Well, the idea of using the parasite Trichuris Suis for converting it into a treatment was coming from Joel Weinstock and his colleagues at Iowa University. And the process was patented by Iowa University, and the university has given us the exclusive right on their patents. So I started to look for partners and went all around the world to make to give presentations of it, and it was I was catching a great deal of attention to this process, but I didn't find anyone really willing to step into it and put a lot of money in it and get into the drug approval process.
Because just quickly on that, initially before you started doing this, Professor John and some of his colleagues, they actually did do a small clinical study, didn't they? With TSO.
Yes.
And if I'm not mistaken, it was very, very effective.
Yes. But this small study was an investigator study, experimental study, for which you don't need full drug approval. So
it was just to really kind of prove the point, wasn't it? To sort of say, look, this thing actually does have huge potential.
Yes, and especially because this was a single center study, it only required to get the permission from the local authorities. And that would have been different if there would be another center outside the state of Iowa, for example. Then the federal agency would have been involved. So because it was just a single center study in Iowa, it was the local authority in Iowa that had to give the approval for running the study, and that's what Iowa got.
And what was the brief summary on the study and the results?
Well, the first study was a very small pilot study where we had, I think it was just 7 patients, and they were given just one shot of TSO eggs. And we saw even on this one shot, we saw a really strong immunological response. So that was very encouraging for the very first initial trial. And then we got permission to run 2 larger trials. The other trials was 50 patients on ulcerative colitis and 50 patients on Crohn's disease. And this study, we were also trying to find out what is the best dosage and what is the best administration period. And so we had one study where the patients were giving 1 shot of TSO every 3 weeks, and the other trial was 1 shot of every TSO every 2 weeks. And
one shot is about 2,500. Is that right?
One shot contained approximately 2,500 eggs of Trichuris. And don't ask how we came to the number of 2,500 eggs. It was just let's see what
thumb in the air kind of thing. Yeah. Fair play.
And it turned out to be perfect. And with the study results, I was able to find one company in Europe, actually in Germany too, which was one of the market leaders in the Crohn's disease and ulcerative colitis diseases, specialized. It was a family-owned company, one of the few very family-owned company. And the owner of the company was a pharmacist and a medical doctor by profession. So he was the only one to decide. If he decided, yes, we're going to do it, everybody has to do it. While in opposite to such family companies, the big pharmaceutical corporations, you have to start at the very bottom. Let's say with the product manager to convince him, then to the marketing manager, then to business development manager, and then to other peoples in the company. You have to work your way up there. That can take a whole lot of time, and it's really annoying. So we were a lucky business company. And started the development. And unfortunately, the owner of the company, he died. And then when an owner of this company like this dies or retires, then it is very different to what it was before. And we had some persons in this company who were very familiar with the entire regulation process. They perfectly knew how to get a drug approved. And the problem was that I think the problem was that the approach later on was how to run a drug approval like a chemical before. But that doesn't fit into what we have in front of us here with the biological organism. So that way they decided, well, a pH value of 2 point something could be way too low. For example, if we would have to treat small children in future, that could be a possibility that the low pH is kind of disturbing in the throat of either small children or other people with certain diseases. So they wanted to raise the pH. And that was the worst mistake we have ever done, because all the studies we conducted before this pH raise was done had very successful outcome. But
at 2.5.
Yes. And the other studies, the bigger studies, the first drug approval study in phase 2B study, where we used the higher pH value, had no significant difference to placebo in the control group.
And they went to a pH of about 5, was that right?
Yeah, that was a pH about 5. And what happened when you raise the pH and put the eggs into the solution, then they start their metabolisms, and they start the process of hatching. And in the bottle where we have the solution there, the drinkable solution, there is nothing to metabolize. So they get very weak.
No.
I made this comment that this might be a bad idea, but they said, well, we tested it. We tested it in that way that we took the pH 5 version and brought the eggs back into the pig. And we saw that in the pig, they started again to shed eggs. So
it's a different host, though.
Yes, exactly. It's a different host. Because even if they are very weak, in the pig, they find food, which they don't find in a human gut.
Yep.
So if they can find food, they can revitalize. And they can then survive and start shedding eggs again. And that's not possible in the human body. So that was one of the reasons why, in my opinion, the big clinical phase 2 study has not shown any significant difference. The other problem we had...
Such a shame.
Yes,
such a shame.
The other problem we had to deal in this big study was that we had a huge media attention, again, because of the exotic approach. It was reported about this in TV magazines and newspapers and journals all over the world. And this is not very good if you are running a clinical study with a placebo control group, because everybody heard about it. And the placebo effect we had in this study was almost 50%. The highest placebo rate that was ever seen in clinical studies.
So people were basically thinking they were getting better.
Yes.
Purely on a placebo. Oh my God.
So people were believing in it. And if you believe in something and you get into clinical study and you believe you've got the agent and not the verum, that's doing something with your body. And that causes the placebo effect.
So was there actually positive benefits from that? Did placebo group?
Yeah, we had a similar effect like the placebo on the outcome with TSO.
Wow, so, that in itself is quite amazing, really.
Yes.
Even if it sucks for the study.
Yes. And at that time, the company, the partner, they decided we dropped the project. That doesn't make sense anymore.
It's such a shame because you were so close.
Yes.
Like, you were so close.
Yes, absolutely.
If they'd just done the pH levels right, do you think it would have been a completely different outcome? Yeah.
Yes.
Yeah. Oh, it's such a shame, isn't it?
It is.
Because you could have cracked it.
Yes.
That says.
Yes, absolutely.
Yeah.
But luckily, when at the time where it was decided to raise the pH value, I had another maggot company in Thailand, and I decided then to start running the project in Thailand as well, but to keep producing in the original pH value.
So you actually had a setup in Thailand, and towards the end of the initial study, when you were like, I assume you were probably like, this is going to fail because they are mucking around with the pH levels. You actually started mirroring.
It was just a gut feeling.
Yeah.
At that time.
But you started doing it in Thailand in sequence almost.
Yes.
But with the original pH of 2.5.
Yes. And it was even more making even more sense because the regulations in Asian country are quite different to the regulation we have in our Western countries. Because Asian countries have a very, very long history in the use of natural medicines, which we don't have here anymore. And that is reflected in their regulations. For example, here in our Western countries, if you have a tea, for example, and you know any kind of tea number X, and you find that this tea has a therapeutic effect on a certain disease. And you say, well, I'm promoting my tea now to be taken for this disease. Then it's a drug, regulatory-wise. And you have to conduct all these clinical studies again, all this toxicity and all this kind of stuff to get a drug approved. In Asian country, they don't care. They have so many natural medicines that the only thing to get them approved by the local Thai FDA, for example, is to have to demonstrate in clinical studies and toxicity studies that it cannot do any harm to humans.
So as long as you can prove that,
yes,
you're good.
Yes, then you fall under the same category, for example, like all the herbal medicines. And then you are accepted from the big drug approval process where you need to do all the expensive clinical trials of phase 2, 3, and 4. You have to conduct a clinical phase 1, showing that it cannot do any harm to humans. That's typically the reason why you conduct a clinical phase 1. And you have to provide all the toxicity studies made on the product. And we had all this, and it was accepted in Thailand, and I got it approved under the category of a herbal medicine or like a probiotic.
Wow. So that makes so much more sense.
Absolutely.
Not only is it cheaper,
Absolutely.
it's so much faster. You know for a fact you're not going to do any harm to someone, but you're able to get something out there that could actually have a huge amount of benefit and save people.
Yes.
It's so much more logical than
and the
than
problem
the Western
here
side.
in our Western countries is you only get the funding for such a very costly approval process if you have patent protection. And the only patent protection we had was one from the Iowa University, and that was on the process of manufacturing. You cannot patent an animal. That's not possible.
Yep.
Animals are not patentable.
Yep.
But the process, how you use the animals, that can be patented. And later on, it turned out there are more processes possible. So this patent is not really a good protection to make an investment of $500 to $1 billion.
Yep.
And
so no one's going to touch it from that regard then. Yep.
But in the meantime, the European regulators, we were able to convince the European regulators that it is not a good idea to put this kind of product on the drug development. It is much better because a couple of years ago, the European regulators created a new category that they call novel food. And the Trichuris Suis perfectly fit under the novel food umbrella because it is taken like a food, it is digested like a food, it is not doing any harm, and therefore it fits under the category. And I applied for the category for novel food, and it was accepted by the European regulators. And
Amazing.
I turned in all the results of the clinical studies we had and turned in all the documentation, all the scientific publications that appeared in the meantime. And the reply was, well, you have everything you need. But what we need to know is that what you still need to do is a large study on healthy volunteers, because a novel food, the major intention of a novel food is to provide it to healthy people.
Yep.
You have all the stuff for sick people, but you don't have a sufficient number of patients treated in a clinical study as healthy volunteers. And here are we again with huge costs to conduct a clinical study of phase 1 with healthy volunteers that required approximately a number of something like 3,000 people.
3,000. And what's the initial cost to do that, phase 1?
Well, normally you can calculate something like $2,000 per patient.
Yeah, so it's a lot of money.
It is.
But imagine if you can get that one over the line as well. That will really open things up, right?
Yes, absolutely. So I'm looking
Which would be amazing.
For somebody who is willing to invest this money for a clinical phase 1 study to complete the novel food application. And then if the novel food application is there, we can even sell it in a supermarket. I mean, there are other probiotics, for example, like Actimel or Yakult. Have you ever heard of it?
Yeah, I have. I definitely have.
Yes.
Yep.
For example, Actimel, it's a huge company behind Actimel. And Actimel for some years ago had the problem with advertising slogan saying Actimel activates your immune system. And they had to stop this because there was no scientific evidence that it does. And TSO has this evidence. So can you imagine if you put TSO into an Actimel, put it in a supermarket, then it is definitely allowed to use the promotion. It activates your immune system because it really does.
That would really scale things up significantly.
Yes.
And have you come across or had much luck so far with trying to find a sponsor to support you in doing this phase one? This is brand new, is it?
Yes.
Yeah. Oh wow, that's amazing. That's pretty exciting. That's really exciting. That is really exciting. So how long have you been approved with the Thai FDA in terms of being able to?
Oh, I have to look it up.
Create product there.
I think that was something around 2006.
Okay, so it's actually been quite a while.
I have to look it up. I'm not good with remembering numbers and dates.
But it's like well and truly more than 10 years.
Oh yes.
That you've. Oh wow. Yeah, that's amazing. and I don't know whether you want to answer this or not, but how many people do you think are taking TSO that you might know of, or that do you have a rough idea?
Yeah, I would say something like 50, 60,000 over the last 20 years.
Globally?
Yes.
Yeah. So it seems like a big number on face value, but actually it's probably quite a small number.
Yes.
When you think of the amount of chronic disease running around the world at the moment.
Especially because all this number of these patients are not patients treated in a clinical study.
100%. Yeah, 100%. Yeah, wow. So 50, 60,000 people. And okay, that's really interesting. So just a little bit back on I wanted to touch on the TSO itself and the process. So when I was talking to Tracy, we were talking about it that you normally re-inoculate every couple of weeks. You take some TSO. Typically, I think people are doing about 2,500 is what you said before, right?
Yes.
So are people seeing significant improvement quite quickly within 8 to 12 weeks?
Yes.
And then over time, they can actually drop the dosage back, i.e. they don't have to take TSO every 2 weeks. Like what's happening there?
Yes. The average time, and that was also shown in the clinical studies at Iowa University, the average time is something, you can see the first noticeable improvements of your disease, at least in Crohn's disease or ulcerative colitis, after taking anywhere between taking dose 4 to 6, which means 8 to 12 weeks after you began. Certainly, this is just an average. It depends on the age of the patient, the severity of the disease when beginning with TSO treatment, and certainly also the kind of disease. So far, we have tested TSO in clinical studies in Crohn's disease and ulcerative colitis, multiple sclerosis. We had some allergy indications, and in autism. And we found very good results in rheumatoid arthritis. And, the autoimmune diseases, the umbrella title of autoimmune diseases, you can find up to 60 different diseases, indications, belonging to the family of autoimmune diseases. We can certainly not test all these at once. I'm happy that we were able to test a few indications in clinical studies for which we got public funding to conduct studies.
Yep. Which is mainly Crohn's and UTI, right?
And MS and autism, for example, and some allergies. Well, you can do a lot of things wrong in conducting a clinical trial if you follow the leads from the chemical drugs. If you set up a protocol that you usually use for chemical drugs, that doesn't work.
Which is what happened to you.
Yeah, that happened, for example, to our first allergy study in Denmark, because it takes quite a long time. Well, short time compared to what other parasites need, but for TSO, 6 to 8 weeks at least to initiate the immune response, beginning of the immune response. And therefore, we said if you want to treat an allergy, season allergies like pollen allergy, for example, you have to begin with the treatment at least 3 months prior to the season, if not a little bit more. What the Danish group did, they started right at the beginning of the season, which is way too short for the product to initiate a immune response, protecting from the pollen allergy. And that's why I have shown on the new website of Televisa company that if somebody wants to start another clinical trial, please contact us first. Follow the lead.
I mean, time frame itself is not typically done very well in these clinical studies, right? They don't normally go over 12 weeks, which in most cases we need to go longer.
Which is understandable. I mean, the reason behind the 12 weeks limitation is that regulators don't want the patient to stay on a placebo for longer than maximum 12 weeks. That's understandable. And some drugs then do a crossover, for example. They do the 12 weeks and then they change the group. Then the placebo group gets the verum and the verum group gets the placebo, which in some cases might work, but not in case of a biological organism like this, because you interrupt the treatment after 12 weeks if you change this, if you do this crossover. So I would say that if you really want to have a seriously well-done study, you need a very much longer term for the study than 12 weeks. And I wouldn't mind to start a study with patients who, for example, are on another medication and then do TSO on top of it. And then at the time point where you can see that the symptoms disappear, taper off the other medication and keep going with TSO. That would be a safe way to approach for the patient. And I think that could be accepted by the authorities. No one has ever done this before. And mostly in most clinical studies, it is kind of a law that you have to have comparable patients. That's why, for example, the multiple sclerosis study took such a long time to finish the recruitment, because especially in multiple sclerosis, it is very, very difficult to get patients that you can directly compare to each other. There are a lot of patients out there, but they have different kind of conditions, different kind of symptoms, and therefore you cannot compare them. And that's why it took such a long time. But on autoimmune diseases, another possible approach for a clinical study where you can combine different kind of indications, technically, regulatory-wise, probably not allowed to combine different indications in one clinical study. But if you start with a very low dose, like multiple chemical syndrome, for example, patients, MCS patients, they react very strong on even the slightest contact with any kind of material. So they do with TSO as well. If they go straight on for a dose of 2,500, that would be way too much for them, which is good for the Crohn's disease and inflammatory bowel disease. But for MCS patients, they need to start with, let's say, 10, 20, 30 eggs maximum, and then step up slowly to a point where they feel good and stay on this point. That would be the optimal approach. But that takes a long time. You need to start low and then stay on the low dose for a couple of weeks and then go a slight step higher and see how you're doing. And when you start getting a side effect, then step back and stay on this dose. And this is
And what sort of side effects are people seeing when they take
Oh,
TSO for the
on
first
MCS
time,
patients,
if
that is
any?
very, very different to all other patients, for example. They can get all kinds of side
So if we
effects.
look at
Yes, yes.
all the other different patients,
The
are
only
they
thing
like people with Crohn's, for
Yeah, the only
example?
thing that can happen on other patients than the high sensible patients, the only thing that can happen
Mm-hmm.
there is you get a slight diarrhea and
Mm-hmm.
slight smelly gut winds for a certain of time.
Yeah.
But the smell is actually a good sign that it works, because the smell is coming
This
from the
works.
bad bacteria. And one thing that TSO is doing first when it comes to the gut, they try to change their environment they are in. It's a natural thing they developed over a million of years. They're trying to change the environment, and that it is a perfectly suitable environment for themselves. And that means that hazardous bacteria or bad bacteria they don't like either. For example, bacteria that blocks the oxygen supply, they would like to have oxygen to survive, even in the gut. But if there are bacteria that block the oxygen supply, we don't know how they do this yet, but they kind of change the gut flora from the domination of bad bacteria to the domination of good bacteria. And during this time of change, the bad bacteria are released from the gut. And that causes this.
Yeah. And how long is that normally lasting for?
Certainly, there is no
Like,
fixed rule for this, but usually something like
It's-
2-3 doses of TSO, something like maximum 6
yeah.
weeks.
And then basically what's happening then is your body and your immune system's getting used to having the
Yes.
TSO there, right? So they adapt to having
Yeah.
them there. Those symptoms
Yes.
disappear. And in an ideal world, those autoimmune symptoms then go into
Yes.
remission, hopefully. And once someone's been doing it for, I don't know, let's say 6 months, can they start to extend the time frame between TSO doses? So they go from 2 weeks to six
Yes,
weeks
that's
to
possible.
8 weeks, for
But
example.
this is
Yeah.
there is no way for me to say you after so and so many weeks you can do this or this.
Yeah.
Because people are
Yeah.
too different. As I said before, it's too
Yep.
difficult to find people that you can directly compare to each other. So it's kind of a trial and error thing.
Yeah.
When
Yeah.
you, for example, you have a disease and strong symptoms, then you start taking a TSO. And if the TSO therapy works, you get rid of your symptoms. Then you have the choice. You
Yep.
can either stop TSO and see how long your remission lasts. And if you get a flare
Mm-hmm.
again, you can start taking TSO again. And usually a
Mm-hmm.
patient who once responds to TSO will respond again, which is not the case on certain
Yep.
other drugs. There are other drugs, if you
Yep.
take them and you stop them and you restart them, it won't work anymore. In TSO, it's
Yep.
not the case. If it worked once, it will work twice or more. And the other option is you can go from the therapeutic dose of, let's say, 2,500 and taper the dose off. Go lower in dosing.
Yep.
And another possibility you have is to extend the period of time of administration. So the three variables are available. And this is different from individual to individual. It's kind of a trial and error.
Yep. And I know again, everyone's so different, and again, a lot of the stuff you've been looking at has been very specific to certain autoimmune, such as Crohn's and whatnot. What's your feel on TSO improving just autoimmune diseases in general? Obviously, there's 60, 70 different types. Do you think TSO is able to benefit all of them? What's your thoughts on that one?
Well,
And again, I know it's a really hard one to answer, but
I strongly believe that there is no wonder drug out there that works on everything.
yeah,
It is. Well, we can see it from the other species that is being used, that they work too. And some species are stronger, like TSO on inflammatory bowel diseases, for example, or on contact allergies, while the Carter americanus DNA is much better working than TSO on diseases like Th2 driven diseases like skin diseases or pollen allergies and stuff like this. There are nuances of different efficacy.
Yeah. Yeah. Okay. That's interesting. It's just it's amazing because you hear these amazing stories from people taking NA, people taking TSO, and in general, most people seem to have a really positive
Yes.
experience by taking helminths, and I think that's quite remarkable. And it's just interesting to see, like maybe you're right, maybe TSO does benefit certain autoimmunes better than others. And I hope one day in the future we get more studies that can kind of show that, because it is really interesting, right? Um, on on that note with TSO, what does it cost to get, like if I wanted to have a crack with some TSO, what would I have to pay to get some? Generally,
For a treatment period of 10 doses, the cost would be something like $1,900 at the moment.
Mm-hmm. And
At,
10
at the
doses,
moment, yes. If,
is
for
it?
example, we would get the novel food approval and can bring the product with a partner company in large quantities, that would come down really, really much.
Yeah. Yeah. So 10 doses would be a dose every 2 weeks.
Yes. 1 dose, 2 weeks
Yeah.
time.
Yeah. 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 No, that's pretty cool. Just
And,
before we wrap it
sorry
up.
to interrupt. And again, these costs are only because we have to conduct all these tests and virology and bacterial tests and have to do all this kind of it certainly like the other species being used out there, they don't do all this. It's just
100%.
If we would do the same without having all this GMP around, that would probably be the most inexpensive treatment available in the market.
Yeah. And I think that's a really good point, because I didn't appreciate until I hopped on and started having this conversation with you. One, how long the process is to actually get TSO and then take them through the cleaning and the setup before you can actually take them. But then two, all the actual the checks along the way to make sure they're ticking all the right boxes from a GMP perspective. And I didn't appreciate that. I had no idea that the process was so lengthy. So it does make a lot of sense that cost is there. And I think that's where we need the scale, right? Because the scale brings the cost down.
Absolutely.
And I'm hoping that we could get you a partner so that we can get this phase 1 study done and we can get them rolled out in Europe, because how amazing would that be? And then you really get some scale behind you, right? And that's when you can really start impacting people positively.
That will be,
especially because which is really
this
cool.
is a kind of treatment that you can even give small children. I mean, all the existing classical drugs out there in the market for autoimmune diseases are pretty bad for small children. And TSO...
Bad for anyone.
Yes. And TSO has been tested in children as young as 15 months old.
Yeah.
And work perfectly.
Have you tested them in children?
Yes,
have you
we had some
before?
University hospitals in different places in the world who make individual therapy trials under the responsibility of the pediatrician in chief of the university hospital, and that worked very well.
Yes, good results, absolutely. Good positive results, not just from a safety perspective, but from a wow, it's remarkable. It's really, really remarkable. Before we just jump off, what do you, from your perspective and all your experience that you've got in the space, because you've got so much experience, what, from your perspective, is missing? How far away are we from having scale and having, helminths like this become a mainstream sort of thing, and people having some positive benefit in their lives that are struggling with autoimmune diseases? How far away is that?
Two possibilities. If I get a partner to conduct the clinical phase 1 trial to complete the novel food treatment, then it is just a matter of 2-3 years maximum. Like, for example, this Actimel company, Danone, is behind this. They would have the resources and the power to run such a study in no time and bring the product into worldwide supermarkets. That will be the perfect number one solution. And the other...
Just quickly on that one. So broadly, it was 3,000 people needed for that study, and approximate cost would be 2,000 per person to complete that study, roughly. Yep.
And the other possibility is compared to what happened with the fecal transplant. Have you ever heard about the fecal transplant?
Yeah, I heard a little bit about that. Yeah, it's quite an interesting
quite an
space.
odd thing too.
It's very odd. Don't tell me you've had your hands in this one as well.
But, on this case, regulators have identified that it is probably not possible to get this running under clinical studies condition and make it a unique and GMP process. And they came to the conclusion that, well, then why not giving us a permission? And fecal transplant are now permitted to be applied officially, approved by the FDA, but they didn't need to go with all the clinical trials and the full approval process. If someday regulators think the same on helminth therapy, they could easily then allow to use it too. Well,
realistically, what is that? Is that something that's realistic in the next 10 years?
I would think yes. I mean, if regulators keep thinking logical, and
it's a big ask. It's a big ask.
Well, you know, some things in nature, and there are much more there in nature that you can convert to a very, very highly effective treatment, don't fit into the existing regulations. We need to modify our regulations so that stuff like this can be approved too under highest possibilities of control. But it's too difficult to find the funding for stuff like this. My perfect solution would be if every country or every larger region would have a committee consisting of high-profile physicians. And if somebody comes up with a treatment similar like this, which cannot be patented, there is no patent protection on it, and therefore there's no way to get all this money to get it approved, then it should be possible to bring this idea to this panel of specialists and let they decide if this is of public interest or not. If it's of public interest, they could use the government-owned hospitals and run clinical trials in these hospitals. So that.
But isn't that part of the problem at the moment? Isn't that part of the problem that with mainstream physicians and GPs and that sort of thing is that they just don't think on this level?
Yes, exactly.
I mean, and that's one of the big problems, right? They would look at something like this and just dismiss it straight away.
For example, like autoimmune diseases. I mean, 60 different diseases under the umbrella of autoimmune diseases. You have any idea
which is
how
ridiculous.
much this
It's absolutely
costs
ridiculous
to a country to treat their people on this? It's huge. And if there is if there is a solution like this, which is not patentable, but you can't use it because you don't find anyone willing to invest the money to get it approved, well, that's a very bad situation, isn't it?
Yes, that's the problem. That's the big problem I see.
If the country would have a committee, a panel, or specialist to look at it and decide, well, this could be of real public interest, we should have it. So let's bring it into our hospitals and make the trials there and get it approved. See what happens if it's really safe, And if we find out on our own cost, on the government cost in the government hospitals, that it's good it's safe to be used, then let's prove it. And that would also bring the cost very much down, because every novelty in drug development has a very high price. And that makes the government suffer too on costs to treat people on very expensive.
Wow. The cost to continue medicating people has got to be so much more than the cost to roll out something novel like this, or at least even try it.
There are so many other really exotic and fantastic things out there. Just one example, there's a sea snail out there. This snail is producing a poison in its body, and it has a tail. When a fish is passing by this cone, it's a cone snail. When the fish is passing by the snail injects the gift, its poison into the fish, and the fish is paralyzed. So the snail has all the time of the world to eat the fish. And the poison that the snail produces is paralyzing the fish under full consciousness. So the nervous system is still intact. For example, if you use this poison as a treatment, injection treatment for people with chronic back pain, what these people are getting now is almost a daily injection against their back pain. And after the injection, they are not allowed to drive a car anymore, for example. And yet they need it every day. If you use this kind of poison from the cone snail, then they might need only one injection per week. And because it's not affecting their nervous system, they can easily drive the car. That will be a nice solution. And another very exotic thing, nature is full of fantastic things. In Asian jungles, where you never can find any hospital or any doctor to treat anything, usually this kind of things are done by monks, having a situation there. And for example, if you get a wound, I found this when I was investigating possible wound treatments. When you get a wound, the wound is open. You need to clean the wound and make a wound closure. And you cannot have material like needle and stitch and close the wound there. They have ants there in the jungle, big ants. And the ants have forceps on their mouth. If you take such an ant and put your thumb on the neck, it opens the forceps. Then you put the skin together, the forceps on the skin, release it, and they close the forceps, and you drill off the head from the ant's body. Then you make a wound clamp. And during the time you do this,
That's strong.
And during the time they do this, the ant is spitting the amino acid they produce into the wound, and that's a disinfectant.
Oh, wow.
It's,
nature.
Yes. I mean, nature is full of things that are really, really amazing.
Wow.
And it's a shame you don't use it.
You could be doing this for.
It's a shame that you're trying all the time to synthesize everything.
Yeah, it's crazy. It's crazy. You could spend lifetimes doing this.
Absolutely.
There's a lot of stuff out there. Right?
What nature can offer. And it's so sad that we are not looking much more into the solutions nature has developed over millions of years' time. It's a shame that we try. I mean, I'm not saying we should only use the natural things. I'm just saying we should use everything we have in a wise manner. There are certainly a lot of situations where you have to take a chemical drug because other things don't work.
Yeah.
But you should not ignore that in earlier stages of the progression of a disease, there are much better solutions than strong chemicals to apply. And for example, placebo effect. Why don't we use the placebo effect intentionally much more? I mean, nothing is more investigated than the placebo effect. Every single clinical study in the world has an arm, a placebo, control arm with placebo. And we always get a placebo effect. And the placebo effect, at least the minimum is 15%. The average is 30%. And as we have seen in our clinical study, it can get up to 50%. I mean, that's
It's absolutely mind
more
blowing.
efficacy than any other drug can offer. Placebo.
That's absolutely
Yes.
mind blowing. If only we could figure out how to tap into
Well,
that
it's
on a
very easy.
conscious
If you
basis.
go
You
to
know,
your
like
doctor
it's,
into the doctor's practice, what the doctor should do is, beside all this kind of medical stuff he normally does, when he hands over a medicine to you that he prescribes you, he should spend more time in convincing you that this is really, really helpful. Do something, anything that affects your brain to believe that this is really good effect for you. Then you're using the advantage of a well developed drug and the placebo. And perfectly, it has to be a combination of natural medicines at a certain time, classical medicines, and placebo. And the combination of the three, that could be the perfect treatment people or doctors should
that could be
apply.
the perfect treatment.
That's what I'm standing.
That would be absolutely, yeah. I mean, the placebo
Yeah,
effect is incredible, and what you're talking about from your studies is mind blowing. I had no idea. That's really interesting. Yeah, that's really
yeah.
interesting. Let's leave it there. We've covered some good stuff. Thank you very much. It's quite remarkable what you've done. Genuinely, really quite remarkable. Your story is incredible. Tracy wasn't wrong when she was telling me that you've got a real good story, and I think it's pretty cool what you've been able to do. Like, you're having a pretty impactful result on people's lives with what you're doing with these TSO, and it's remarkable. And I haven't tried it yet, and I think once I've given the NA a bit of a time, I'd like to have a crack with it as well. So I will be having a try at some point. But thank you for what you're doing. I think there's thousands and thousands of people out there that are benefiting
My
from what you're
pleasure.
doing, and that's pretty special.
It's very
It's pretty
rewarding
unique.
for myself too, because if you have a job
It's
that you really, really love and see results
yeah.
like this, how better can it be?
It's genuinely amazing, and I think there's probably so many people out there you don't even know the positive impact you've had on their lives, which is pretty special. And I really, really, really hope that you can get that phase 1 study done, and you can roll this out on a grander scale. And I will do what I can in the background and whatever to try and help get that money raised for you, because I think it's quite an amazing thing what you're trying to do. And just genuinely from my own perspective, thank you very
My
much.
pleasure.
And on behalf of everyone
Anytime.
else that's out there,
Thank
thank
you.
you. It's amazing.
I promise I just keep
And
going on.
just thank. Yeah, well, that's good. I mean, we need more people like that out there, right? That are just getting on and doing these amazing things, unsung, right? Like, I had no idea this sort of stuff was happening, and if it wasn't for Tracy, I wouldn't have known. So it's awesome, and it's been a real pleasure sitting down and having a chat with you about it, because I've learned a lot, and you've opened my eyes on TSO and also this whole world of natural therapy, probiotics, and TSO itself and what it can do. So yeah, thank you for that, and thank you for spending the time. It's much
Thank you very
appreciated.
much. My pleasure.
Yeah, cool. You
You have
take
a
care,
good day.
and I'll chat to you again soon. Thanks, Detlev.